So my name is Elizabeth Ross. You all know me as Betsy, which is great. I'm completely amazed at this group. You are calling your children heroes, but I have to tell you, parents, you are my heroes for coming here and coming so far from places as far as Slovenia, the south of France, Scotland, places as exotic as California, not to mention New Jersey. And it's really a wonderful time to meet your children, to hear your stories, to understand more about what it means to have these rearrangements in 8P. I didn't bring my biography, unfortunately. I was bad, I'm sorry, so I'll just introduce myself. I went to medical school here at Weill Cornell and did my residency in neurology at Mass General in Boston. I did 2 postdoctoral fellowships after that, one in genetics at Harvard and one at Rockefeller University here, and then went out to the wilds of Minnesota to build my laboratory in basic research, and came back here about 15 years ago to Weill Cornell, to my roots. So over the last, I'm ashamed to admit it, nearly 30 years, I've been working on finding genes that cause rare diseases in children and adults that affect the nervous system, with the idea that we want to learn more about how the brain is constructed and how it functions, and along the way, hopefully find new avenues to treatments. So I also have the honor and privilege of directing the Center for Neurogenetics and Laboratory of Neurogenetics and Development here at Weill Cornell. I usually show these slides at the end of a talk, but I want to introduce some of the team to you so you can understand that all of this kind of work takes a very large village. And in the basic studies that I'm going to show you today, these are investigators from my laboratory who actually generated the data that you'll see. There are about 15 members in the lab, but they include assistant professors, postdoctoral fellows, graduate students. These two, Raphael and you, just finished their PhD thesis work with me this last month, and wonderful technicians. And we have collaborators not only across the globe, but here in New York at Sloan Kettering, at NYU, at Columbia, at New York State Psychiatric Institute, at Cornell, University of Michigan, and now new studies starting up with Dr. Chung here on chromosome 8P. In the Center for Neurogenetics, we have a large team of genetic counselors. Shanila Lakhani is here today. Devora Siegel, you've just heard from, who's in the neurogenetics research clinics with us. We see both children and adults. Mary Vo is an adult neurologist who sees patients with us on Fridays. And for bioinformatics, we have computer scientists who are working on the genetic material that we collect. Paul Walujewicz, in particular, is interested in structural variants, so he'll be working on the rearrangements that we see in your children. And Bojena is leading our biobanking and primary cells, so she establishes the fibroblast cultures that we then reprogram into neurons for the investigations that you'll see. So 8P is a major challenge, as I'm sure you know from the experience that you've had so far, but each of your children has a unique story of changes in 8P, and they have a unique developmental journey. So we need to find the common threads in that story and the smallest region on 8P that is shared by all of them to get a sense of what may be the most important genes to focus on, to see what avenues we may have moving forward. I love the way Rebecca put it earlier today, to make sure that your children can be the best themselves they can be. So with these understanding of the changes, the deletions, duplications, inversions on chromosome 8, they encompass many genes, and our goal is to find one or several that are responsible for the most important features of your children's clinical picture. So this is just one of the deletions of one of your children here in the room on 8p, and this is the list of genes that are found in that region. As you can see, it's quite a list, and some of them are familiar to us as geneticists, like DLGAP2, CLIN8, and RGF10, MCPH1, and CSMD1 we're just learning something about, but many of them have names that really don't mean anything to us yet because we don't really know what all of these genes do. Several of them, like MCPH1 and CLIN8, cause disorders when there's a mutation in that single gene, and it's a recessive disorder if you get 2 copies of that mutation. There are problems that we understand, like CLIN8 is associated with neuronal ceroid lipofuscinosis, and MCPH1 is associated with a primary microcephaly, or small brain phenotype. But that's not what we're going to see when they're involved in an 8p rearrangement, because it only involves 1 chromosome in your children. So there's only going to be 1 copy that's going to be affected, and it may not actually have any impact at all on the processes that we know these genes can affect. So it's going to be really quite a puzzle to figure out what's going on. So knowledge of the genes that are confirmed to contribute to this condition may prove useful to find currently available treatments, and I'll give you a couple of hints about how that might work, and further research into the mechanisms and how those genes function will give us clues toward new treatments. So we're just at the beginning. What does the road look like? The phenotype, which are the history and the symptoms of your children and their stories. Some of you have already donated skin cells that we can then reprogram into early embryonic cells and then bring forward into neurons or other cell types that should be investigated, and those are planned. From that kind of information, we'll be able to combine information about the gene structure around the 8p region, and we can select candidate genes. We have a few that we're interested in right now, but the more we know about the types of changes that your children have, the more we can narrow the regions in. When we take a look at how the cells behave in culture, we'll have more of a clue about which genes might be really important. Then we go from there to gene editing in induced pluripotent stem cells, which I'll tell you a little bit more about here. And then from there, we can make animal models. We can look at models in, believe it or not, worms. And we can also look at zebrafish, which are a very convenient experimental model. We can also create models in mouse, which can give us a clue into behavior. And from there, we'll understand some of the cellular mechanisms and some of the behavioral mechanisms. And after all of that work, we can start thinking about molecular modeling and drug screening in vitro, and then in animals. And then we can go to the FDA looking for approval to go ahead with clinical trials. Clinical trials then go through a number of phases, 1, 2, and 3, before you actually arrive at a treatment. But we're here at phenotype and gene structure, so we're really at the beginning. And we're just starting to investigate reprogrammed cells and planning out ways of doing some gene editing experiments. So it's really going to be a very long road. Our hope is that as we go along, we'll pick up some clues that may be useful along the way. So we don't actually have to get all the way down to a phase 1, 2, or 3 trial. There may be useful things along the way that will be helpful to physicians as we work together. So the data that you're going to see here has nothing to do with chromosome 8p. I am just showing you some examples of how genes can be investigated to be able to get insights into how they work and what the impact is of changes that we see. And I hope it's going to stimulate some discussion. One of the things that we do in the Center for Neurogenetics is when we have a patient that we've seen in clinic who has a variant in a gene that has been associated with their condition, it may be a mutation that's never been seen before. So we can't say for sure that this change is causing the problem that we're seeing in our patient. So then it's called a variant of uncertain significance, or a VUS. What we can do then is take that mutation into the lab and see what kind of effect it has on the function of that protein that the gene encodes. And so this is an example of a couple that we've looked at in children who have epilepsy and behavioral problems that are associated with how the synapse functions. And the synapse is the connections between neurons and the way they signal with each other. So oh great. So in this patient, there's a single nucleotide change in a receptor called NR2B, and that is part of a glutamate receptor that gets excited by neurotransmitter. And the second patient has a mutation in a syntaxin binding protein, type 1. So syntaxin is part of something called a snare complex that helps bring vesicles within the neuron to the membrane so that it can be released in the synapse and cells can communicate. So these are when you have disruptions in these two genes, these are two different ways that you can end up having communication problems between nerves and end up generating epilepsy. So in these two cases, what we've done is taken fibroblasts from the skin, which some of you have donated from your children, and we have reprogrammed them using a set of viruses that take the fibroblasts back to an embryonic state. That means the fibroblasts, the reprogrammed cells, iPSCs, can be differentiated forward into different types of cells. So we could make cardiac muscle from that. We could make liver cells from that. We can make neurons from that. And in this case, we have made neurons. And put them together into a differentiation protocol that lets the cells aggregate. They come together and form little balls of cells, and they create what we call organoids. And we do that by giving them different types of factors that embryonic cells normally need in order to differentiate into a brain cell. After a while, they start recapitulating some of the events of embryonic brain formation. Very rudimentary steps. So these will never make a person. These will never make a true brain, but they will just follow some of the steps that are involved in brain differentiation. And these are pictures of organoids that are stained for early genes that are involved in creating these neurons. So we can also take neural progenitor cells from these iPSCs and put them into culture so that they differentiate into neurons that are reaching out to each other and forming networks, and they're signaling. And each of those dots that you see in both pictures are spots where there are synapses forming, connections between nerve cells. And these cells have been plated onto an MEA plate called a micro electrode array, multi-electrode array plate. And each of those electrodes will pick up any cells that are right on top of the electrode and firing. So when we do that, we can actually look electrically at how these cells are forming networks in a dish. And here we've got cultures that have been made from wild-type healthy cells, from children cells who have a mutation in STXBP1, and in this case, a child with a mutation in SNAP25. These were made from fibroblasts that Dr. Chung sent to us a few months ago, and these NR2B cells. And when we look at how they fire in a dish, you can see that compared to wild type, the STXBP1 cells are actually not firing quite as much, and the ones with the SNAP25 and NR2B mutations are more rapidly firing. So this suggests to us that indeed the variants that we see in these three patients are having an effect on how these proteins work and how neurons will function in the dish. And we can do something called raster plots to actually look at how these wells in the plate are firing. So each one of these plots is looking at the firing at one electrode, and we can look at the patterns. So you can see that the SNAP25 cells are firing much more in terms of their firing rate. The NR2B cells are more often bursting, and the network percentage is increased, as well as the synchrony firing together rates, while the STXBP1 mutation is less active. So what does that do for us? For one, it tells us that these changes in the gene are actually having an impact on function. But we can also use the cultures that we have to expose the cells to different drugs to see how that's going to affect their function. And in this particular series, we found that a receptor antagonist, memantine, will actually normalize the activity of the NR2B mutant neurons in culture. So this means that as this child has behavioral problems going forward, one of the drugs we might consider using is memantine, which you may or may not know is currently being used for the treatment of Alzheimer's and memory disorders. So we wouldn't normally think of using that medication for this child, but it gives us a clue that this might be a good thing to try. So we can also learn more about the mechanisms of pathogenesis in deleterious mutations. And in the particular experiments, we tried to look at the function of a protein called occludin, which was something that we found to be mutated in a family that we encountered from the Middle East in Doha. They had 2 children with a condition of microcephaly, small brain, and polymicrogyria. So I don't show a normal comparison here, but I think you can see that the brain is not very well formed. There's also a problem of leaky vessels that leads to calcification within the brain. And the mutation that we found in occludin is actually a protein that's very important for tight junctions between cells to make barrier properties in things like blood vessels. This is a recessive mutation. It was necessary to have 2 copies of the mutant occludin in order to express the problem. But we were curious about this because there are mutations in other tight junction proteins like JAM3 that lead to cerebral calcification, just like our family, but they don't actually show microcephaly and diffuse polymicrogyria. So that suggests to us that there's something different about the function of occludin than most tight junction proteins. So we decided to take a look at that, and Raphael Bendriam in my lab started by looking at the localization of the protein, and he noticed that not only is occludin found in tight junctions between cells in early brain in the mouse, but it's also in centrosomes that are concentrated at the ventricular surface, and that was unexpected. So we found in looking at proteins that attach to different parts of the cell when you break it up and you put it into a centrifuge to see whether it's at the membrane or it's within the cell or at the centrosome, that it's at the centrosome as well as the plasma membrane. Okay, so now I'm going to turn you all into developmental neuroscientists and tell you about a very important event in very early brain development, and that's a particular progenitor cell for all neurons that's called a radial glial cell. And when the differentiation of cortex is reaching a point that radial glial cells are present, they have to divide in a particular way that's very restricted. They have to make a symmetric division, that is that compared to the surface of the ventricle, they have to have a vertical cleavage plane so that you get a completely similar daughter cell on either side of a division. So with a vertical cleavage plane, you can get 2 daughter cells that are going to go back into the cell cycle and divide again. That's how you get the size of the ventricle in very early stages. If, for example, you get a horizontal cleavage plane, you get a different outcome from a cell division. One of the daughter cells will go back into the cell cycle and divide again, but the other one will leave the cell cycle and become a neuron, okay? If you have a skewed cleavage plane, you can still have a tendency to develop, one cell that goes back into the cycle, one neuron. And these vertical cleavage planes are really, really important in very early development. So in fact, when cells are dividing at embryonic day 11 and 12 in the mouse, almost 100% of those cells have a vertical cleavage plane. And if you don't, you're not going to end up with as many progenitors to be able to end up with neurons. Okay, so why did I go through all this? If we look at the versions of occludin that get made by this gene, there's a long form, which is the wild type, and there's a short form that lacks a part called exon 3. And the patients that we found with these mutations had an interruption right there in exon 3. So you would think that it would completely knock out that gene. But it doesn't, because there's a short version that doesn't start until farther down in the gene, and that still gets expressed, okay? So we decided to model this in human embryonic stem cells, and to do that we used CRISPR-Cas9, and I know some of the parents here were asking about that, so I'm just going to take a minute to describe it. Cas9 is an enzyme that was found in bacteria that is sort of a really rudimentary start at making an immune system. So Cas9's job in certain bacteria is to recognize foreign DNA and chew it up. Okay? So some very clever scientists decided to co-opt this property and to create a system so that you could select where Cas9 goes in the genome by using a guide RNA. Sorry, can I go back? By using a guide RNA that will bring Cas9 to a particular sequence in the genome and cut the gene. Okay? Having cut the gene, both strands of it, the cells will then try to repair it. And when they do that, they'll leave out a few nucleotides, which is enough usually to keep the protein from being made. So that's what we did. It's called non-homologous end joining. It's just stitching things back together, but it's not a perfect stitch. And when you do that, we are able to see that we only make the short form of the protein of occludin, and it's going to be found at centrosomes. And without going into all of the different permutations, we have different antibodies that will only detect the C-terminal end of the protein. That won't distinguish between the short and the long forms. Another antibody that's just at the N-terminus. And when we look at our programmed cells, they are making only the short form at centrosomes, not the long form that goes to the membranes between cells. So what does that do? We then took the iPSCs and we made organoids, which you've heard referred to as mini brains. And this is what they look like at day 20 and day 40 in vitro. And if you look at the wild type, they are significantly larger than the mutated versions. And if you look at their growth curves, essentially, the diameter of each of those mini brains, you can see that the ones that bear the mutation are quite a bit smaller. That says to us one of two things has to be happening. Either the cells are not dividing very well, or they're dying, okay? So we can look at that, and we can use markers. It doesn't really matter today what markers those are, but we see that they're not proliferating very well, and they're getting hung up in one phase of the cell cycle, in M phase, which is where that cleavage plane is going to make a lot of difference. But they're also dying more, so they label for apoptosis. And they also are coming out of the cell cycle more rapidly, forming neurons sooner than they should. And then, very importantly, they're depleting prematurely a type of glial cell, radioglial cell, that expresses a gene called HOPX. And this is a cell that is only made in human brain. It's not in the mouse. So if we didn't look at human ES cells, we wouldn't know about this particular cell type having problems. Also importantly, organoids don't have blood vessels. So we know that the effects that we're seeing on cells are primary to the function of this form of occludin. It's not because of the blood vessels. So when we look at the microtubules, the spindle apparatus that actually pulls the chromosomes into each daughter cell when it's about to divide, there are problems there. It's not very well organized in the mutation, and also at the poles of the microtubules, called the astral spindles, there are problems. And the reason that that is important is because these astral spindles actually help pull the chromosomes apart during division. And so if you have a problem of contact with those astral spindles, you're going to get a skewing of the cleavage plane, and the cells are going to go out of cycle faster, and they may die. So this is probably one of the main reasons that we're seeing the proliferation problems in occludin. And this also has an impact on how the cells, the chromosomes themselves, get pulled apart. So you may see here that there are some that are not really well lined up, and so it could lead to a condition called aneuploidy, when you get an extra chromosome in one of the daughter cells or one fewer in the other. And that's probably why the cells that we see dying are taken out. So this is the model of how it's working. In the mutant context, we see sort of a slight disorganization of the chromosomes. They're not getting pulled apart, and this is just because these cells lack full-length occludin. So one last example that I want to show you today is a gene called LIS1. The reason that I think it's relevant here is that LIS1 was found in patients because of a large contiguous gene deletion syndrome on chromosome 17. It's not chromosome 8. And it was called Miller-Dieker syndrome. And it leads to a complex syndrome, but the most important feature is a brain malformation called lissencephaly, or smooth brain. And you can see a lissencephalic brain next to a normal MRI. And children with Miller-Dieker syndrome have a similar facial appearance. After you've worked with this condition, you can actually spot a child across the room who probably has MDS. And there may be other organ involvement, sometimes kidney problems, for example. But through genetic studies and collecting families, working on mapping the gene, it was also found that you can have this kind of lissencephaly with a single point mutation in the LIS1 gene. So the LIS1 gene is the one gene in that whole chromosome 17 region that's responsible for this brain malformation. And that has led to a lot of work that my laboratory and others around the world have done on this gene over time. The kinds of things that we can do are to isolate neurons from a genetically altered mouse that has a knockout of LIS1, and we can show that lissencephaly is thought to be a problem with neuronal migration. Neurons are first born around the ventricle, and they have to migrate out to the cortex to form the thinking part of the brain. And when they don't do that very well, the cortex is disorganized and it's thickened, which is what you see in LIS1. But making a mouse that has one of the LIS1 genes inactivated, you can then isolate neurons in a dish and see how they migrate. And if you look at, compared to the average speed of migration of a wild-type neuron, in the dark bar, you can see that a mutant neuron doesn't move as fast in the dish. And you can look at things like D-serine that will further boost that activity, and that the mutant neuron doesn't respond. Now, D-serine activates that NMDA receptor that I was telling you about, the glutamate receptor. And so we thought, well, maybe there's a problem with calcium entry into the cells, which is the major part of what happens when you have a neurotransmitter that sticks onto a receptor. But that's perfectly fine in these mutant cells. What we did find was that the next step in the process, activation of GTPases, is abnormal when you're missing one copy, just one copy of this LIS1 gene. So our model is that in the inactive state, before a signal comes in, you have all of these proteins that will come together after a neurotransmitter hits the end process. And LIS1 is a very important part of this complex that helps the cytoskeleton reorganize itself so that you can then have these protrusions that come out. This is going to be really important in situations like axon outgrowth, which is something that may contribute to things like agenesis of the corpus callosum. It's going to have an impact on how nerve cells form synapses to communicate, and a number of other processes. So LIS1 is important in that. That told us that there might be an issue with how synapses form when you disrupt the amount of LIS1 that's present at the neuron. And that's because when synapses form, these protrusions come out of the neurite of the nerve, and they sort of explore the territory. And when they find a target that they like, they turn into a synaptic spine, and that's where the connection is going to be made. So in these experiments, we took the LIS1 deficient mouse, and with Wenbiao Gan at NYU, we were able to make a cranial window and use a 2-photon microscope to actually look at the synaptic spines in the cortex. And the thing that's great about this is that we can take, an hour's recording, and we can come back 48 hours later and look again and see if those spines are still there. Or we can look weeks later or even months later to see whether we can still see the same spines that we saw forming way back at the beginning. And what we've found with this LIS1 deficiency is that there's a problem with synapse formation, but once those synapses form, there's a problem with them being eliminated. And the elimination is something that is important for synaptic plasticity. We can also culture these neurons in a dish and look at the process cell by cell. And we can inactivate the gene in a particular brain region. In this case, we've done it just in the hippocampus, which is the place where memories are formed. Once we've done that, we can use electrical recording in the hippocampus and see how things like LTP, long-term potentiation, which is a molecular mechanism of memories, work. They don't work when you don't have LIS1. And we can use behavioral studies in the mouse to see how they remember things and what you can do to improve that. So this is a classical conditioning model, which I can tell you all about later, but I think I want to leave some time for questions. There are also certain types of glia in the brain. They're named for the Greek for glue. They're support cells that are really important for synaptic turnover. And we've found in these models that there is an overactivity of microglia, that we can actually use certain drugs to be able to reverse that. So some of the long-term effects of having the deficiency of LIS1 can be counteracted. So I just want to leave you with a few thoughts that this is really an amazing time in science, that we can look very deeply into the processes that are causing, problems in brain function. And this is going to have a tremendous impact on our ability to fundamentally understand what the problems are underlying chromosome 8p structural variation. And our hope is that as we're going on this journey, we'll have clues along the way that will help us make sure that your children reach all of the potential that they have. And I thank you so much for being our partners in this. And I'll leave the floor open for just a couple of questions.
Yeah.
Thank you.
You guys, we have time for one question before we do the breakouts. Does anybody have a question? You're going to have a chance to speak with Dr. Ross in the breakouts. Okay.
Great. No, we have one? Okay.
Of course, now I can't find that. Here we go.
It's okay.
It doesn't matter.
All right.
Oh, because you made a breakout.
Heck yeah.
Hello?
Yeah.
So, if we get to the point where we find certain medicine works or helps and things progress with our children, is there an expectation that they have hit a certain ceiling at this point and things would just, keep them from, or we be able to progress beyond that ceiling of where, creating new neurons and things like that? Or is it kind of a cap?
Great question. So there are windows of opportunity in the life cycle. So for sure, it's not going to be possible for us to effect complete cures for a number of reasons, both because of the developmental course that the nervous system takes, and also because in 8P conditions, there's almost certainly more than one gene that's involved in all of the symptoms. Having said that, if we find something that works really well for synaptic function, it may be really helpful for seizure control if that's a problem. There may be behavioral issues that we could improve. I don't want to be so neurocentric that I give you the impression that brain is the only thing that's going on. I'm sure that there are other issues as well. But we'll learn from differentiating these cells about many different organ systems. There'll be opportunities for looking at specific problems. That's one reason that it's really important for us to know what are the issues that are most concerning to you and to the quality of life that your child has.
Got it. Thanks.
Once again, let's thank Dr. Ross for coming and spending the day with us today. So now we have a special treat for you guys. We've separated you guys into breakout groups. If you have a color tag on your badge, take a look to see what room you're going to go to. They're all on the second lower level. So where you went to pick up your lunch, that elevator bank, just go down one more. For those of you that don't have